All right. All right, I'm going to call it back to order. Our next speaker is online. Carrie Pike would have been here if we had done this a year ago. But Carrie took a new position in Indiana. I don't know. I want to say a few months ago, but Carrie, you can maybe correct me. It seems like a year ago. Wow. Carrie, go ahead and say something. Let's make sure we can hear you.
Hi, can you folks hear me?
My fault. Just a sec. I'm going to turn that down. Carrie, you're coming through just great, but a little too loud.
What else is new?
Yeah. All right. And there may be some feedback if I keep my mic live. In any case, Carrie's got 15 minutes, very short time, which is why I was eager to get us going. She's going to talk a little bit about genetics in jack pine. And without further rambling, I'll hand the floor. Oh, actually there is, oh no, I've got that covered. I'm going to hand the floor to Carrie. Carrie, you're on.
All right, I hope I'm not blasting you away. Good morning, everyone. I'm sorry I couldn't attend. My title is a little funky. We're going to segue a bit from the direct impacts of a fire for a moment and consider some of the long-term impacts that fire regime might have on the genetic structure of jack pine. And this is important because it'll have ramifications for selecting seed sources for planting and assisted migration efforts in the future. So John, I hope you don't mind. I borrowed this from the 2011 Inconvenient Truth 1 talk that you had given at the Cloquet Forestry Center as part of the jack pine symposium. And the point here is that jack pine has experienced notable range contractions and expansion. So it's proven to be a fairly nimble species over time. And Minnesota is a great place, but it's a relatively small geographic area compared to the continental-wide range of jack pine. It's also relatively flat, although I live in Indiana, which is flatter. And yet, based on phenotype, 3 of 8 subpopulations of jack pine were found to reside in this relatively small geographic area. That means almost half of all the phenotypic, not genetic, but phenotypic diversity of the species exists in this relatively small area. And this was determined by Roland Schoenike's work in the 1970s. And what this poor guy did, if you look at this map, I don't know if you can see all of these little circles and spaces, all these little dots on the map, but each dot represents a collection area where Schoenike sampled 10 trees from each of these sites, and he collected needles, cones, a bark plug, and an increment core from each of these trees. And maximizing the computer resources of the 1970s using cards that they stuck into computers, I don't know what that was about, he conducted a cluster analysis to cluster these samples based on their phenotype. It's a multivariate analysis. And based on that, he classified these 8 different populations. And you can kind of sort of see, it's not great, but if you go up to like BC and to the northwest, and then there's this line that runs down to Alberta to the western interior. So what's interesting, though, is that he identified these 3 different subpopulations occurring in Minnesota, the north central, middle, and the south central, which is an awful lot. It's a very steep gradient to have in one small geographic area. So based on Schoenike's work helped to inform this map that you saw in the Native Plant Communities book that John Allmendinger helped write. So we have these floristic regions that John discussed. I'm not going to talk about the southern or the northwestern, but I'm going to focus on the green and the blue in the middle, the difference between the north and the central. So these regions were developed based in part of Schoenike's work, undoubtedly some of John's work, paleohistory of jack pine and its cohorts. So Schoenike identified, looking at the cones, he associated closed cones, so those serotinous cones that are good after stand replacing fires, with cones that were curved in the northern area. I don't know if cone curvature has any adaptive quality, but certainly serotiny does. And in the southern part, in the central and southern areas, jack pine cones were observed to typically be straight in stature and open to take advantage of the cooler prairie fires. Now, I have pictures of straight cones that are closed and of curved cones that are open. There's been a lot of mixing on the topic, but he associated these 2 types. So one of the questions as a geneticist, I asked what is the underlying cause of this population structure that Schoenike unveiled, and one of the most obvious ones is the historic fire regime that it likely influenced it. But also as a geneticist, and some of you studied evolution, the question I have is, if fire goes away, shouldn't this population structure, how come pollen from east to west isn't mixing and breaking down these population barriers? Because pollen can move, pollen will come out in Brainerd, but unfortunately by the time it gets over to Carleton County, the female cones aren't receptive yet. So we think that these lines, these population barriers that we have, may be reinforced by differences in phenology from east to west that are driven by heat, temperature, and precipitation gradients. Enter in the gall rust. Now I'm talking about gall rust because it's a big problem in parts of the state. This is a map, another poor resolution map, but it was from a paper by Dietrich et al. One of the et al. Authors is Dr. Bob Blanchette, who's down at the University of Minnesota. This is in the Canadian Journal, and they went around and sampled gall rust from across the state. And so there's two genera in the state. There's Endocrinartium hortensii, which I believe is the pine, and Crinartium querculum on jack pine, and the Crinartium one is the pine oak. The closed circles in the top right and the northeast are the pine gall rust, so there's not much oak. And in the southern parts in Hubbard and Cass County, that's the pine oak, which is considerably more virulent. Interesting, though, that there is this odd line that goes across the state. This kind of goes right through Itasca County, somewhere around Lower Red Lake down into Carleton County. And if you take this map and you put this same line on it, you can see that it's interesting that the line that separates the gall rust populations also roughly coincides with the difference between the northern and the FDC populations, which is kind of interesting because Schoenike did not have the benefit of Dietrich et al.'s work. And Schoenike, when he sampled, actually deliberately did not look at gall rust. He ignored it. Tree had it wasn't sampled. So it's almost like a natural coincidence. So we don't really, we believe that there's some underlying cause for this, something under the bark that may be going on. As a natural resource manager, you might ask, can we capitalize this? Is it possible that jack pine populations in the central area may have some local resistance or tolerance to the local gall rust and the same in the north? So the typical gall rust we observe, the pine gall rust, and I will report on these plantings in a minute. In the northern part of the pine gall rust, these infections occurred at fairly low frequency. So we would find the gall, it would appear in a main stem, and the tree would flop over, as in this picture. So they're usually lethal, but they're very low frequency, so not a big problem. In the pine oak, this is down near just south of Brainerd, you would see multiple galls across the whole crown of the tree on the main stem. They were lethal. They were ugly and very, very high frequency. So it was a much bigger problem down there. So what I'm going to report to you on is we measured several traits on a full-sib progeny test at 2 sites. What's interesting is that I collected this data, even though none of this, we never intended to look at cone serotiny. So I don't know the cone traits of the original parents. I don't know the cone traits of the grandparents, but we do know the latitude and longitude, and we can identify whether it came from the north or the south central area of each maternal and paternal grandparent or line. So we're on our second generation. So I'm going to describe the second generation progeny, which was created from a controlled pollination from a first generation orchard, which originated from open pollinated seed from a grandparent tree that was wild tree out in the woods. We maintain all this information. The one good thing is that we tended to put the first generation orchards tended to be near the wild tree, so there wasn't a lot of movement from wild tree to the first generation. So we have this really neat progeny test. It's located at 2 sites. We had 18 trees per family, which is pretty good replication, and 100 families, which is great. A randomized complete block. So what I did, I assigned the floristic region. Because it's full-sib, I know the mother and the father, and we assign the floristic region to the mother and the father of each tree based on the grandparent's origin. We don't know if the grandparent trees had open or closed cones, and we don't know if they were straight or curved, but they likely didn't have any gall rust on them. And I find breeding values for all these traits to each family. And I'm going to show you that. So here are the location of second-generation populations, one in central St. Louis County, one in Crow Wing County down on the Morrison County line. Results. So heritability. Heritability is a number between 0 and 1. It tells you the likelihood that if you look at one tree, in this case, the likelihood that the other full-sibs, its full-sibs, are going to have the same types of traits. A really high heritability, like in the case of stem straightness and cone serotiny, means if one tree has closed cones, its other full-sibs are most likely also going to have closed cones. It tells you how similar, metric of similarity. Tree volume I put on just to contrast tends to be lower. There's a lot of heterogeneity on these sites, so that's expected. Stem straightness was pretty high. So if a tree was straight, had very good form, wasn't wiggly, the chances are good that its full-sibs also were straight. Cone stature and serotiny were very heritable, and rust incidence was extremely heritable. That was the highest one. And note that the standard errors are small, so these are fairly reliable. So I did this discriminant analysis. The discriminant analysis is a type of multivariate. And basically, I wanted to assign each family to its maternal and paternal floristic region and see if the traits follow the expected trends that Roland Schoenike had observed. In other words, are closed curved cones associated with FDN still, like 2 generations later, are open straight cones associated with FDC? And at the FDC site, where pine oak gall was prevalent, are families from that FDN more likely to be affected by the disease? So I'll just give you a quick result. There were no significant associations of maternal lineage, so I couldn't track it, associate them by the mother, which surprised me. However, when I looked at the pollen parent, the paternal lineage, we did find significant associations, and I'll show you what that looked like. So I'm not going to show you numbers. This is basically a picture that represents what the actual data showed me. So this is the summary of the discriminant analysis. This is at the Brainerd site where the pressure for the gall rust is very high. And this is classifying each family by its pollen parent. And differences that I will unveil to you from left to right as you go across are different. If you go up and down on the y-axis, they weren't different. So let me show you what it looks like. So basically, the cloud of points for these traits did separate according to the FDC and the FDN of the original grandparent tree. So those are going from left to right. Those were different. And they were different in this sense. Straight cones were associated with the left side, so the FDC, and the FDC families, pollen parents, were also associated with a low disease incidence. Now, serotiny didn't behave, and maybe it's because this isn't the mother lineage, and maybe because I'm making this data do something it wasn't intended to do. The closed cones were towards the top, open cones towards the bottom. That second axis, the y-axis, wasn't significantly different. So it didn't help. We couldn't classify them by serotiny. And the FDN, though, was associated with the opposite, the high disease incidence and the curved cones. So the result follows Schoenike's predictions. That the central area had straight cones, and in our case, we were interested in disease incidence, and they had a low disease incidence. But when you brought the northern sources down, they tended to have curved cones, and they also suffer from a higher disease incidence. Now, you have a slide in your PowerPoint that summarizes these results, but I didn't have time. So what do we draw from this? One, it supports the hypothesis that Schoenike's population structures likely have a genetic basis. This could be confirmed with population genetics. But we have to do more research to investigate the inheritance of cone and other adaptive traits. It would be fairly straightforward to study whether serotiny and cone stature comes from the mother or the father. This data set, I made it work for this. So that needs to be linked. That needs to be studied further. But cone traits in jack pine may be linked to gall rust and other traits in jack pine. This has been suggested as this terrible word in genetics called linkage disequilibrium, which means there are traits that are stacked together that tend to stick together. But we have to be careful how we interpret it. It doesn't mean that a tree that has open straight cones is going to be inherently resistant to gall rust. It just means it has a higher probability of having some resistance than a tree that is the other type. Implications for seed zones: we may need to consider floristic seed zones as opposed to the existing seed zones for jack pine because of the sensitivity of transfer. But if you're planting trees and you're having problems with gall rust, you may want to consider using seed orchard seed instead of wild seed, because we've done selections to improve it, to reduce the incidence of gall rust in our populations. And then out looking kind of bigger scale, there are implications for assisted migration. If we're going to continue to move oaks north, as has been proposed, oak being considered one of those winning or species, then we may be at risk for moving the pine oak gall with it, in which case we may also need to consider moving jack pine. So there are implications, long-term implications of moving seed, particularly of moving jack pine, that could affect our ability, or the ability for them to adapt in a future climate. So that's all I have. I'm going to just put my acknowledgement stuff there. I got to thank the MTIC and all the folks in Crow Wing, St. Louis, and IRRB, DNR, and the U of M. So that's it. That's my 15.
All right, Carrie, thank you. That was terrific. I want to thank the audience here for bearing with the AV stuff. We're